Welcome to New York Bio's virtual breakfast series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Sam Waksal, serial founder of companies like ImClone, Cadman, Mira Gene Therapies, and the newly formed Equilibre Biopharmaceuticals.
All right. Good morning, everyone. My name is Jennifer Hawks Bland, and I am the CEO at New York Bio. And we are very excited that you are here with us for what is our final 2021 virtual breakfast series. We have had an amazing group of guests over the course of this year. And frankly, who knew when we started having these conversations back in 2020 as a result of the pandemic that we would be able to have such an amazing reach and engagement with you all as we've continued these conversations. I dare I say I think this is probably something that will stick coming through the pandemic. So we have a special thank you to our sponsor this month, Iser Amper, and we've really appreciated their engagement and sponsorship. But today, for our final conversation, we are joined by Dr. Sam Waksal, and we are going to talk all things company creation, company formation, financing, and the New York biotech ecosystem. I think that Sam's long and storied career, we can probably talk about just about anything related to biotech. We're thrilled he's joining us, and I will turn it over to Derek to do the introduction. Thanks.
Thanks so much, Jennifer, and good morning, Sam. It's so great to have you here. Usually when we kick off, there's a particular story, there's a particular company, there's a particular thing that one of our guests did that's kind of the easy intro. Although much like Jennifer said, I think we could probably pick out one thing from each decade, if you will, with you. We could probably spend the entire hour. So usually we tend to kick these off with a bit of an origin story for each of our guests. And given that could probably take the hour, it would be really great to kind of wind back the clock to think about, what was it like when you first, left academia and started ImClone. For the edification of the audience, Sam is someone who has, founded multiple biotech companies, and more importantly, somebody who has brought, by my count, at least 4, if not more, drugs all the way from discovery to patients, which I think is about the most important accomplishment you can have in our industry. And Sam, if I missed one in there, please correct me, and I apologize for being short-sighted on that one. So great to have you here.
Well, thank you very much. It's a pleasure. So there were 5.
I knew I was going to miss one.
No. So the great thing about the origin story is that I was an immunologist. I was at Mount Sinai building an immunology program for the medical school, and it was a great time. I had come from Boston, and I really looked at the world in a different way through the eyes of an academic, and working on getting grants, publishing papers, and just being part of a world that I thought was terribly exciting. And I was working as an advisor to the new emerging field of biotechnology. Remember, Genentech went public in 1980. The world was changing. We were cloning genes. We were making monoclonal antibodies. And there was a new world out there. Sometime in the middle of the 1980s, I decided to found a biotech company, and with colleagues that were really smart academics. I didn't really get the essence of what it would mean to build a real biotech company. And when I began, the first thing I did was try to find space. And I lived in New York City. So I wasn't going to move. And when I started to look for space, I had one real estate agent, as it were, drive me through the Holland Tunnel and on the way to a space that was owned by Grand Island Biological. And when we got there, it was a beautiful space. We were looking at their labs. They had left that space, and they were empty labs all ready for a new tenant. and somebody else might have taken it, I asked, so you mean I have to drive through the Holland Tunnel and drive for 40 minutes on the freeway and get off of an exit? And this is where I said, I can't do that. Isn't there anything in New York? So indeed, on the way back, he said, you know, there is a building on Hudson Street. In fact, on Varick Street, because there was another biotech company that was on Hudson Street, actually. And he said, it's actually space that's great. It's a shoe manufacturing company. They make plastic shoes in a printing building. Remember, we used to print all of those things before we went public. And I went there. It was great space. And I said, I want this space. He goes, well, you're going to, and you should all know that it was $6 a square foot a year, not a month, a year. So I said, done. I'm taking, he said, well, you're going to have to, it's the only asset of this bankrupt company. You have to go to the courthouse this week and bid on it. So I went to the courthouse with a lawyer, and the owner of the building was there. And the judge asked who we were, and the owner told him, he said, you can't bid. So he said to me, go ahead, bid. I didn't know what to do. So I said, $5,000. And he said, okay, that's not enough. Eventually, I bought the lease, I think, for $25,000. We'd already raised some money. And the rest was that we built ImClone there and got more space in the building. The man was actually a wonderful man and invested later in ImClone, Joe Rosenblatt, a unique and wonderful human being. And actually, when I later founded Cadman, I tried to go back to that building, but they had leased it to NYU. And what was really wonderful was that we got to build ImClone there. And I was with recently a wonderful man, Ken Lipper, who was at the time deputy mayor. And he helped me get an industrial development bond for the space, the equipment. I never had to use any equity to build out the space. So it was a great time. And we started ImClone. And my view was, this is going to be easy. I'm going to build a biotech company. How
could you think there was no precedent, right? Other than like Genentech, was there any precedent for, you know, was there a path?
Well, there wasn't, and that was the great thing. It was the thinking was I could do just about anything I wanted. Yep. And we worked in a multitude of areas. So whereas now I hear investors say, well, you can just work on one thing, right? Just one thing. I worked on 20 things. And that's why we got so many drugs on the market and later. And what was interesting is we did diagnostics, we did vaccines, we worked on antibodies, we worked on small molecules, we worked on everything. And we did a deal with Lilly on diagnostics. We did a deal with Abbott later on diagnostics. We did a deal with Letterly in vaccines. And we made interesting therapeutic products. So it was new. And at the same time, the organization that became NewYorkBIO was inchoate and just thinking about what it was going to be. And Jack Hutner, who was the first executive director, came to me and said, we want to build an industry organization in New York. What do we do? I said, well, first of all, we have to find other people in the industry, which was in and of itself challenging since everything was new. And
well, what was the environment like? How many of them were there?
Probably about 2, if I remember correctly, but more grew.
They found you and the other guys.
Yes. At least
Right.
it was easy to find meeting space at that point.
That was easy. And Jack wanted to figure out how to create an organization that could represent this industry that we felt was going to grow. I mean, New York is great. People don't realize that for biotech, you've got Rockefeller, Columbia, Mount Sinai, NYU, Cornell, and more. And so what it did for us is really build the infrastructure of science, right? We had science in New York. And in fact, people also don't realize that the second amount of NIH funding, this really important asset to the industry of science, came to New York. And so we had a lot of great science in New York. I was able to hire scientists without a problem. People who wanted to stay in New York, people who wanted to help create a new ecosystem of what became biotechnology.
It's interesting, you know, you saying it that way. I remember ages ago, I was at a place called the New York Academy of Sciences. This is probably 13, 14 years ago. And we had a number of kind of focus groups on, you know, why doesn't New York produce more startup companies? How can New York be better in terms of an industry environment? And, you know, Larry Whitty, for the audience, who was the head of R&D at ImClone for many years, would talk about how when they hired people at ImClone, you hired people that wanted to be in New York City. You hired people that wanted to stay in New York City, because for a long time, people would wave their hands and say the cost of living is too high. And I thought one of the really interesting things was, you know, people want to live here. And, you know, one of the great ways to build a culture around that is to hire people that want to live here or work here.
No, absolutely. And people forget that the cost of living in Cambridge, Massachusetts, or in South San Francisco is a lot.
Yeah.
And you know, Larry was great. I hired Larry when he was working at Columbia. And I hired people away from Genentech, which was unique. We brought in our head of biologics, became somebody named Dr. Zhenping Zhu, and ZZ, as I called him, was unique and really made with me all of those monoclonal antibodies that became great drugs and that are on the market, helping so many people with different kinds of oncological disease. And he was my head of biologics. And then when ImClone was sold to Eli Lilly years and years later, we went public in 1991. And I talked about two interesting targets, FLK1, FLK1, which became VEGF receptor and is now Cyrenza. And I talked about a stem cell molecule, which we thought was unique. And I talked about vaccines. Later, really just a year later, we made what became Erbitux. And John Mendelsohn at the time had just come to Memorial Sloan Kettering to be chief of medicine. He worked, he was, when he was at University of San Diego, University of California at San Diego Medical School, had worked on EGF receptor. So we worked on that. And later, John became the head of MD Anderson and was on ImClone's board of directors. And he died a couple of years ago of glioblastoma. And it was devastating to a lot of us that knew how important he was to the field of cancer. So all of these things were new. You got to do things that no one had thought of doing before because it was so early. And I'm excited that we did all the things that we did at ImClone. I don't know if venture capitalists would have paid for all of that right now. They're never smart enough to do that. And I made sure that I said that. They're never smart to do that. And there were those of us that were smart enough. So it was great. Zenping. I mean, Larry continued with ImClone and then on with me at Cadmon for a long time. Zenping, when ImClone was sold, went to Novartis and became their head of biologics. And then he came back to me at Cadmon and now is the chief scientific officer for a huge public Chinese company making a group of great drugs, 3S Bio. So it began to populate an ecosystem other than ImClone. And that's what's important in the ecosystem in New York. People go off, they begin to do other things, they do new things. I remember that Sharon Mates, who is now the CEO of Intracellular, was living down in the Washington, D.C. area. And I got her to come up to New York to do a new genomics company. And so she became a critical part of the New York ecosystem. So it is not easy, but it isn't that hard. The big problem is when a scientific discovery is made at Rockefeller or at Columbia, or remember, we have 2 Nobel laureates at Columbia in Eric Kandel and Richard Axel, who are uniquely brilliant men and have done a lot of things in the field of biotech as well. And we have Siddhartha Mukherjee, who's doing new things. We had Lou Cantley at Cornell, and he's gone back.
We just lost him.
Yeah, that was too bad, but he's got a new biotech company, which is quite interesting with Sid Mukherjee. So the world now does stuff here. A lot of the venture capitalists, because they believe that the leadership exists up in Cambridge or in other places, tend to take the asset, the value of the patent, the idea, the product, and they move it. I don't.
And we're thankful for that.
No, I think that's changing a little bit. I think over the last, I don't know, probably about 8 years ago, personally, I thought the tide started to turn when Versant and the Accelerator Corp put teams here and actually started looking under, the rocks and turning over stones in academia here and being willing to start companies here. And, we've seen that grow. We've seen that grow more and more. And you thankfully have started all of your companies here, which is fantastic. But, we can't count on you to start all of the companies.
Oh, you could. I've started a lot more than
many of them, but you can't start. We can't start all. If we're going to really increase the number of things that we put out, we do have to probably have other people start some companies too. But I think that was when the tide probably really started to turn. But as you said, we've had great science here for an incredibly long time. I'm actually really interested to understand how you think things have changed from either from an entrepreneurial perspective or from a financing perspective. I can imagine, ImClone, was Wild West, and I could only imagine how you kind of got people to invest in ImClone originally, because you can say the word biotech, I would imagine most of them didn't know what it was. But then even, when you started Cadman, it was basically during the nuclear winter for biotech venture funding. So, until recently, it's unclear to me that you've started a company at a time that was easy to find financing.
Well, biotech is an interesting field. I was speaking to an investor recently, and he said, I don't understand the language. And, that's a fascinating thing. We're talking about science. We're talking about products that hit a unique target of signaling. We're talking about products that affect pathways that are made with small molecules, made with antibodies. And it's a unique field, because I could start an app tomorrow and get it on the market in a couple of days. I can't do that with biotechnology products. I need to get it through regulatory authorities. I need to finance it over and over again. And that isn't easy. So I think while the environment has changed, I must say the number of venture capitalists that one was speaking to in 1985 when we started MClone and opened our doors down on 180 Varick Street in 1986. It was the wild, wild west. That no longer is true. And now you have, instead of one or two or three venture capitalists who are willing to invest in biotech, you have dozens and dozens. And it changes the world for everyone. When I left academia, I felt naked. You asked how I felt. I felt naked. You know, I had been going to conferences, immunology meetings, cancer meetings, and spoken about data. And I was an academic. And I thought to myself, oh my God, what are people going to say? And it was actually interesting because a couple of people, including my brother at the time, said to me, do you know what they're going to say? When you get a drug on the market, they're going to say, wow, because they aren't doing that. And it's true. In academia, we just didn't do that. We made a discovery and talked about it. I remember working on a recombinant murine leukemia virus. I was able to talk about that for at least 5 years. Same thing, over and over, publishing nuances of descriptions. And that's something that in biotech just isn't useful. Now, you make a product, you've got to move forward, and you've got to do it with in a time frame that makes sense for investors and makes sense for the world. And COVID has taught us that we can do it a lot faster. I mean, look, at Pfizer, they made an antiviral, and they went from discovery to getting it on the market in about 1.5 years. So what did it teach us? It taught us two things. Number one, it taught us that we say things that just aren't true. It doesn't take us 5 years to discover something. It takes us very little time, and we have to do it in a time frame that makes sense. Yeah. And since I've just started so many biotech companies, I now know that number one, I've got to do it faster, because since I was around probably before you were, when you were in diapers and I was starting MClone, it means that I finally realized how fast we can do things. Right. And so it is irrelevant to me when somebody says to me, well, we can do this in 1 year. And that means we can probably do it in a few months. We just have to be tough and thoughtful and insightful and do a lot of things that heretofore we never did.
Sam, going back to the academia and not recycling, but sort of adjusting your findings and republishing, do you think that academia is starting to realize the value of the entrepreneurial students that they have, and trying to encourage that more as you're hiring out of academia today versus 20 years ago?
That's, it's such an interesting question, because I have two views about academia. I think that discoveries can be made, they have no clue what they mean. So, when I first dealt with licensing officers, VPs of research at academic institutions, I used to smile at what they said they wanted and what they deserved, because things are changing in that area as well. I must say, now I don't smile, now I laugh. I think that academia has an important role, and we need to have more NIH funding, and we need to do great science, because their world is to do great scientific discoveries. I don't think it's their job to create drugs. I don't think it's their job to understand how a target is important from the point of view of clinical development. I think it's the job of the biopharmaceutical industry. I also think, in 1986 and 87, as we were thinking about how to build companies, people didn't, the pharmaceutical industry didn't even know what a monoclonal antibody was.
Right.
Now, we all do the same thing. There is no difference in platforms for a biotech company or a pharmaceutical company. We all do the same thing. So the difference is, I think, in point of view and speed and understanding why we have to leapfrog certain areas of science and development. And we're doing it. I think that in New York, we have these new opportunities. The more new companies that are created, the more future companies there will be.
Right.
Yeah, I think so. And if you think about it, the kind of things that you did with ImClone basically laid the foundation for, basically the people at NYU who took over the space and turned it into an incubator that would now foster more companies. So I do want to spend some time talking about your new company, which is Equilibre. And you're working in an area that has been classically extremely difficult from a biology perspective. So can you talk a little bit about how the company came to be and what the mission is and how you're approaching things?
So I started the company during COVID. Imagine, nobody was working. I was working. And what's quite interesting is that I never find an area difficult. And I say that in a glib way, but I believe that my job has always been, this is me. I've tried to connect dots, and I look at areas of science that are unmet medical needs. In China right now, there are probably 30 anti-PD-1 antibodies in development, maybe 40, maybe 50. Anti-PD-1s, do we really need that? It's absurd. In this country, we're doing the same. It really makes no difference. We like to be part of a paradigm. And being part of a paradigm, we know in science, it's how science moves. Arthur Kornberg at Stanford, when I was at Stanford, used to tell me that he'd publish a single paper in the field of a DNA replication. And then there would be 100 or more papers published later, a facet of what he had done. So we like to live in paradigms. Science moves, though, anti-paradigmatically. And I try to think that way. If 10 people are working on something, I don't want to work on it. It's just not interesting, and probably not even as important as after that first approach was made. So for me, the field of neuroscience is sort of the last stronghold of metaphysics in medicine. It's really a cool area. And in every one of the areas that we can think about from a disease point of view, what's important is that there aren't real drugs for them. So whether it's Parkinson's or seizure disorders or traumatic brain injury or frontal temporal dementia or Alzheimer's, we need new drugs. Right now in America, we spend about $300 billion a year literally wiping the mouths of Alzheimer's patients. And in a few years, that number is going to be a couple of trillion if we don't find a drug. So it is up to us, both in academic approaches as well as in biopharmaceuticals, to find new drugs in this area. Equilibre, it's from the French, équilibre, means to balance. And we're sort of trying to rebalance what are the normal events in the brain. The sequelae of a lot of those events, whether it's a genetic event, a viral infection, a mechanistic a trauma event, a bacteria, makes no difference. There are a lot of single events in a spot in the brain. But then there are the sequelae of those events, and whether there are changes in how neurotransmission takes place, all the way to immunological events, inflammatory events that we have to figure out how to modify without the severe side effects that we see when we use a lot of drugs that we try and use right now. And with Equilibre, we've started working on a drug that is already moving toward phase 3, and I think is unique, one, and two, has a great side effect profile, and three, it's working, it's interesting, and it's important. And that's what I do. And I don't get baffled by the fact that everybody thinks the areas are tough. It just makes them more interesting for me. Makes them interesting for me scientifically. It makes them interesting for me from the point of view of development. How we, meet those regulatory hurdles that the FDA challenges us with all the time. And we're doing New York.
Yeah, I think one of the biggest, go ahead, Jennifer.
Oh, I was going to say, given your history of working on a whole bunch of things at once, how with Equilibre, are you narrowing down sort of the types of conditions you're focusing on to start? Or are you boiling the ocean here as well?
That's a great question. We're trying to get a drug approved, and so we're doing it with, excuse me, a real focus, and at the same time trying to figure out a lot of other indications. And at the same time, I'm doing other things, other drugs, other things. So I can't just do one thing. That's not who I am. That's who some finance people are, and that's who lawyers are. Lawyers do that really well. I know we've got a lot of law firms that are members of New York Bio. I'm always amused by legal points of view. How's that? How's that for being nice?
Yes, and as a recovering litigator myself, I appreciate your politeness.
It is. I want you to know how rare it is for me to be polite.
Well, let it be said, on December 14th, Sam Waksal was polite on camera nonetheless.
So I think one of the biggest challenges in a lot of ways in neuroscience has been, do we understand the biology or not, right? It's difficult to actually understand a drug when we're not quite sure what the downstream biology is going to look like. I think that's probably been one of the reasons that Alzheimer's has been such a difficult disease to get a hold on. Not only that, because the readout takes so long, right? So when you think about where we are now in terms of kind of the field of neuroscience and how well we understand or how poorly we understand certain conditions, how does that type of, we'll say, macro understanding feed into either how you thought about Equilibre or how you think about other areas to go into?
That's a really important piece of everything, to understand the essence of something, whether it's a pathway or a human being. One of the things in science that most influenced me, I think, was Francis Crick one day, for sure before you were born, was giving a lecture, and it was talking about the right-handedness of DNA. And I didn't know why he was working on that. We all knew that DNA was a right-handed double helix in nature. And after the lecture, I walked up and I was speaking to him, and he was explaining to me why he did the experiment. It was because somebody had published a stupid paper and he was disproving it. But then he said, then he told me how he thought about it. And he said, one day I made myself very small and I got inside the double helix. And he said, I looked about me, and he said, what I noticed is that a right-handed double helix had an aesthetic. The left-handed double helix didn't. And so I knew that the right-handed double helix was real. And he said, you will learn, pointed his finger at me. Francis Crick was bloody brilliant, right? And he said, you will learn that in science, in medicine, and even in the crass world of economics, if it doesn't have an aesthetic, it's not true. And I finished talking to him and I'm walking away and I was thinking, was that real? Is that true? And it took me a while and I learned it was. I also learned that the way we had to imagine things, the way we had to connect dots had to be unique, and it had to be the way Francis talked about it. We had to make ourselves very small and get inside of a cancer cells, the way I thought about Erbitux initially, and get inside a cancer cell and figure out how signaling takes place. We are learning new things about neuroscience, and the paradigm shift is difficult. Everyone thinks that A beta sheets in the brain are it. They aren't. And for example, both at Mira Gene Therapy and in their collaboration with colleagues like Greg Petsko, who is one of the more brilliant structural biologists on the planet, and in collaboration with Scott Small, we worked on gene therapies that became unique because we understood it wasn't the A beta sheet. If that was like asking somebody who was looking for keys under a lamppost why he was looking there, and he said, because the light's better. It's not where the keys are. And so I find neuroscience fascinating. I look at it differently. I think about it now differently. It's one of the first areas I worked on. I made a rabbit antibody to mouse brain, and I was working on neuro events, and I found out that my antibody looked at T-cell differentiation antigens, and I said, it's easier to be an immunologist. But the point is that neuro is fascinating, and we're getting it now. And there are very smart people out there, and there are very smart ideas. And I think that my team has great ideas now and knows how to deal with things that are going to push the field forward. And thus far, I haven't been wrong about my science. And so I think I'm right about my biopharmaceutical approach.
Well, we were fortunate enough to have Zandy as a panelist during our annual meeting. We had a gene therapy panel, which was dynamite. And as I said to people beforehand, the prep calls for that panel were just as good, if not better, than the public discussion. We were really fortunate. The panelists were terrific, and we spent really a couple hours in the week before talking about where gene therapy was going as a field. And all of these folks are working in genuinely difficult areas. If I remember correctly, all of them, I believe, were not only doing gene therapy, but were doing so for disorders of the brain, disorders of the CNS. So it's an area, I think, where you've now attempted to tackle neuroscience twice from two directions.
Well, it's interesting. The gene therapy field is tough. Genetic medicine is a new area, and Mira was a spin out of Cadma. And Alexandria Forbes is probably the best CEO in that field and is doing unbelievable stuff. And I think over this next period of time, people are going to go, wow, it's transformative. And that's what we are meant to do. Our jobs in biotech, if it's one thing that everybody should listen to right now, this is my view, our jobs in biotech are to change the world of medicine. That's our job. It's in agriculture as well. It's a little easier. But our job is to do that, whether with vaccines. Look what happened with COVID. We literally, science changed the world. And I wrote a chapter for a book that Jeremy Levin had edited. And my chapter was My chapter was, it's the science, stupid. And that's true. And the science that we do will change the world for us. This is really an age of biotech. I believe we will change the world, whether it's with things, whether it's with epidemics, pandemics like COVID. We didn't know in 1919, something that killed, maybe even up to 100 million people, that the influenza vaccine, the great influenza vaccine, now there's a vaccine. Yeah. We didn't know what the virus was, and people were dying on trains going from Philadelphia to St. Louis, and it was 18 to 35-year-olds. Pandemics, cancer, Alzheimer's, all will be literally changed as far as outcome by our industry. And biopharmaceuticals can be proud that it's trying to do all of that. And we're meant to do it not by being Me Too and not by sitting there in the pool of paradigms, but rather being anti-paradigmatic and figuring out how things really work and how we can alter outcome.
Sam, we've got a couple of questions from the audience, and I almost hesitate to ask this, but I'm going to anyway, since you're being polite today. So the question is, what do you think about the Edge-at-Home controversy, and will it hurt R&D or commercial opportunity in Alzheimer's disease going forward?
It is a difficult question. I generally believe that when things move forward in a field, they teach us something new and let us move other things forward. And the Edge-at-Home, that drug and the work that Biogen did was really important. It moved things forward and taught us perhaps that the FDA could approve something for neuro the way it did in cancer by looking at a reduction in the size of plaques in the brain, and that might be important. I think that Al Sandrock and the team at Biogen did a unique job in getting it approved. Getting attacked because it was approved was stupid on the part of any of us. Even though colleagues of mine like Greg Petsko and Scott Small believed that it wasn't the right target. It's not the etiology of the disease. That might be true. But then that was the issue of the agency. It is the FDA's role to approve a drug, and drugs have to be safe and efficacious, right? That's the goal. And was it safe? Risk reward. We're learning more right now. Do I think it's a great drug? Probably not. Do I think it was a mistake on the part of the agency? Maybe. I don't know yet. I'll have to see what happens moving forward, and we'll learn things. We learned, for example, with Novartis's now expansion of the SMA market, spinal muscular atrophy. We now have thousands of patients that have been treated with that gene therapy. What we're learning is the side effect profiles are not benign, and it's difficult. But we learn a lot more when a drug gets on the market. We have real world data. So I didn't answer your question because I'm being polite. And a part of me way back when felt that drug shouldn't have been approved by the agency. And then when I sit around and think about it, I think maybe it should have, because we're going to learn in real world analysis what it does to patients when it's used earlier and earlier. The fact is that this drug is going to be used by some people, not most people, because most neurologists are going to say, we don't want to use this drug. It just doesn't work. The data doesn't tell us that. But somebody's going to take it that's in a certain area of their Alzheimer's disease progression, and we're going to learn something from that. And when we do, it's going to teach us either what not to do or what to do going forward. I don't think that's a bad thing.
Can we shift gears to finance a little bit?
Sure.
We have a question from Dennis Purcell. And he asked, what do you think about the amount of capital that has entered the sector in the last few years? And he also says, congrats on your success. And full disclosure, Dennis is also a New York Bio board member.
And full disclosure, Dennis Purcell is somebody that's worked with me for many years when he was at H&Q and a banker and later. And Dennis is a friend and I love him dearly. And he gets this field. He's financed it. He's represented companies. He's done IPOs, and he's been a big investor from a private equity point of view. I'll tell you, there is more money now than anybody can think of. Is that a good thing? Probably not. More companies are financed, and I am of the school that when one does that and just spreads money around, you're just wasting money. I think that a lot of the companies that have started, as I look at them, venture-backed companies, I laugh. I laugh because I believe, and that's out of pure arrogance on my part, because I believe, and Dennis knows, how badly behaved I can be sometimes. I believe that we have far too much money and we just spread it around to things that if one venture capitalist thinks an area is great, 10 other ones will think the same thing because the one thought it. And the one was probably wrong. And so we have a lot more money than we need. Back in the old days of science, the NIH went through periods where it didn't have a lot of grant money. Yeah. It still funded the same good stuff. So the bad things never got funded. Bad things not getting funded is good. Funding yet a 10th CAR T company is not a plus for society. Funding a 20th CAR T company, if 10 wasn't a plus, is even less of a plus. So I'm not of the school that there's all this money, but that's okay. I mean, people get funded, companies fail, and that's, okay from an economic point of view. When General Electric isn't the same company that it was years before, and when Tesla starts an explosive new area in the car industry, that's good. Things fail and new things arise, and that's progress.
Yeah, it's like you said before, you learn from one thing to another, and perhaps one of the silver linings here may be, well, if we fund, another 30 or 40 PD-1 companies, perhaps, a few of them will learn something about immunology, or a few of them will think about ways to do it differently.
No, they won't.
You start to do things theoretically, one might learn ways to do it differently, and then next time they take an angle that is not, PD-1 or PDL1, but actually goes now push the boundary in a different direction.
That's true.
I think the whole
that's true, but that's academia's job as well. We are trying to find out in the field of immunotherapy for cancer what things truly synergize to give us more than that 20% response rate that we get in patients with non-small cell lung cancer. But you're right. It is, this is a field where we learn from our mistakes.
I mean, in most everyone, right? So are you supportive then of increased, when I was in DC at GSK and then at Merck, we obviously pushed really hard to increase funding for NIH and basic research, right? For across the US. Do you think that academia needs more funding for the basic research to answer some of those basic questions? Or is it still, is it just too much across the board?
No, I think that NIH funding needs to increase. I think science in America needs to grow. It is out of academia that the biotech industry arose. We cloned genes at Stanford and UCSF. We made monoclonal antibodies in Cambridge, England, and at Wistar in Philadelphia. It is an area that needs to be funded. We need to make sure we have more NIH funding. It is, I have to tell you, I was on CNBC once and they asked me, would you give up funding for the environment for more NIH funding? And I said glibly and got in trouble in a New York Minute. And I didn't mean, funding to fix the environment. I meant funding to understand environmental science. And, a lot of it, I think, is just not important research. Then they asked me if I wanted to give up monies for the National Endowment of the Arts and the Humanities for more NIH funding. I said, never, never. And they said, why? And I said, that's because it makes us human. So we need more money for discoveries. We need more money for science, but we also need more money for the arts and the humanities. We need to be insightful about our new world, and our world's complicated now. It's much more complicated than it was in 1980 and infinitely more complicated than it was at the turn of the century, the last century, the 20th century.
One of the things that I noticed in looking, I went and dug around a little bit. And one of the things that's consistent across all of your companies now is they all have a statement on representation in clinical trials and the ways that you actually look to get your therapies towards a really diverse set of patients. And I think that's one way that the industry is changing, I think, for the better, in thinking about the kinds of patients that we can enroll in clinical trials and what our clinical trials look like within each disease state.
I think that's critical. I think biotech's been a leader in that. The question of diversity in America is important. We are a country that in the 1950s, if you looked at TV shows, they were a bunch of suburban white people. We live in a diverse country, and our clinical trials need to be that way. A lot of times clinical trials were done and there were no women in the trials. It's stunning to me that things like that occurred, but they did. It was the times. I don't get mad about what happened during a period of time because it was part of that time. What I get worried about is that we don't do the right thing now. And it is up to us to make sure that we have in our clinical studies, in our companies, on our boards, a diverse population of individuals. That means that you need boards with women and people of color. We went through periods of time when boards had no women, no people of color, when we had C-suites. I like that. I learned a term a while ago, a C-suite, whatever that was. I'm joking, but we had C-suites that had guys that they didn't even look like me. They were really, just old white guys. And that's a bad thing. So now we've become more diverse, not just in our clinical trials, but in our companies. We're doing better things, not great yet, but better things. I'm going to say that at the same time, you don't do it just for that reason, right? You don't create diversity and grab somebody from, and so, I can't get in trouble. My family will kill me. You don't just create diversity for diversity's sake. Excellence in science, in finance, in companies is what the goal is. And you do that because you can go out there and find great people that are great, not because they're women or people of color or a lot of other things, but because they're great. And we have to move that forward. We have to do clinical trials that are real world trials that really answer questions about disease, the way the disease occurs in society.
Well, and one thing that I think New York has in spades is diversity, right? Particularly when you look at the diverse population for clinical trials. I think that's one thing that makes New York so attractive for companies, particularly looking today, right, to do clinical trials on a diverse population.
Yeah, I mean, but look, New York institutions are able to do clinical trials. You can't just do clinical trials in places like New York. You need to do them also in Kansas, in Ohio. I grew up in Dayton, Ohio. The Wright brothers were from Dayton, greatest innovators ever. I believe that the world is a diverse place, and clinical trials need to be in multiple places so that we can say it wasn't one institution. But New York is great. It's why New York's so interesting is that we're a small, Manhattan's very cool. It's a straight up city, right? We make very tall buildings, but we also have everybody living on top of each other. And it has, what it does is create for us an understanding of reality. Reality is an important thing. I ignore it too often, but it's very important.
So you had mentioned the book from Jeremy before. I'm actually going to put this into the chat, and I encourage people to look at it. It's a not-for-profit book, and it's a handful of essays from a number of leaders across the biotech industry. Important here is I'm trying to remember when it came out, but it couldn't have been more than a month or two into the pandemic, and it's thoughts from really terrific people in the industry. I can only imagine that, one of the great things over the past, 20 months was the speed at which everyone in our industry responded to COVID. And Sam, I can imagine that, Jeremy called you in the morning on a Monday and said, I'm going to put this together. We need your thoughts. And I can imagine you got your section to him on Tuesday. How did all of that, as we only have a couple minutes left, but I would love to hear just how that came together quickly, and just generally the leadership of Jeremy and everyone else in the industry.
Well, Jeremy's a unique individual, and he was the CEO. His first real job was me making him CEO of an M-clone spin-out, Cadis Pharmaceuticals, and Jeremy's had a great career. He called me and said, I'm doing this. It's really important. Can you write a chapter? And I got him the chapter in a couple of days. The book came out in the fall of 2020, and he did a unique job there, as he's done in other areas for bio nationally. And so we did that, and it's a great book. Everybody should read it. There's some great chapters in it. The best one is mine.
I just want to bet with myself. Sam,
on that note,
wonderful guest. Yeah, on that
yes,
note.
On that note, Sam says he's the best. Sam, thank you so much. Your insight and your thoughts around the industry and company building have been fantastic. But more importantly, it was a very enjoyable hour, certainly for us and for our guests. So everyone, we are taking a hiatus for the holidays, and we will see you back on Tuesday, January 4th for our next virtual breakfast. Everyone, have a good day. Thanks.
Have a good one. Thank you so much, Sam.
Bye-bye.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.